Many more bacteria produce greenhouse gases than previously thought, according to a new study. Caltech researchers discovered a new class of enzymes that enable a myriad of bacteria to breathe nitrate when in low oxygen conditions. While this is an evolutionary advantage for bacterial survival, the process produces a greenhouse gas, nitrous oxide, N2O, as a byproduct, the third most potent greenhouse gas after carbon dioxide and methane. However, unlike carbon dioxide, nitrous oxide is not long lived in the atmosphere, meaning that any interventions to curb its emission can have immediate benefits. For example, overuse of fertilizer for crops provides soil bacteria with abundant nitrates, which they then convert into nitrous oxide. More judicious application of fertilizer could both cut down on greenhouse gas emissions and save farmers money. Nitrous oxide is a much more difficult greenhouse gas to monitor than carbon dioxide, but with this research we now know that there are way more sources producing nitrous oxide than previously thought, says Woody Fischer, professor of geobiology and senior investigator on the new study. Understanding where and when this gas is released into the atmosphere can help us make smarter decisions. There's a not too distant future in which a farmer has information about the communities of microbes present in their soil, enabling informed decisions about how and when to use fertilizer for landscape health. A paper describing the research appeared on June 20th in the journal Proceedings of the National Academy of Sciences, led by former postdoctorate scholar Ranjani Murali and principal investigator James Hemp. The team examined the genomic sequences of tens of thousands of different microbial species throughout various environments on Earth. Most cells in the biosphere utilize certain proteins called reductases to breathe or respire oxygen, but Murali and her team discovered a wide swath of reductases that have evolved closely related proteins to respire nitric oxide, producing nitrous oxide in the process. Nitric oxide and the nitrous oxide are intermediate chemicals produced during denitrification, the process by which bacteria break down nitrate, the chemical found in fertilizer. Bacteria are able to switch from respiring oxygen into nitric oxide in many different environments, wetlands, alpine soils, lakes, and so on, when oxygen level starts to drop below approximately 10% of atmospheric levels. We've missed large regions of the biosphere where nitrous oxide was being processed because these proteins were undiscovered, Fischer says, and now we can much more accurately predict through genomic sequence information which organisms in which environments are producing nitrous oxide. There are way more than we thought, she said. Geobiologists had previously believed that anaerobic pathways like nitrate respiration evolutionary came from before the ability to breathe oxygen in our early single-celled ancestors. This study flips the script, according to Fischer, demonstrating that the proteins that enable nitrate respiration actually evolved from those that respire oxygen 2 billion years ago. Microbiologists often predict that metabolism's microbes are capable of performing based on comparative genomics, explains co-author James Hemp, a former Caltech postdoctorate scholar now of the company Melioria Bio in Utah. However, these hypotheses are rarely tested experimentally. Our work has dramatically increased the biochemical diversity of one of the most studied enzyme families in microbiology. This should serve as a warning that automated metabolic analysis without experimental verification can lead to incorrect conclusions of the functions of microbes and communities. Murali, now faculty member at University of Nevada, Las Vegas, is the study's first author. In addition to Murali, Fischer and Hemp, Caltech co-authors are former graduate students L. M. Ward, now of Smith College, and Usha F. Langappa, PhD 2021, now of UC Berkeley, and additional co-authors are Laura A. Pace, Robert A. Sanford and Robert Jennis of University of Illinois, Urbana-Champaign. This is from California Institute of Technology, Caltech, on phys.org. Please leave your comments and thank you for your support. Finally, support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.